That's a load of science.
Okay. Hello, everyone, and welcome to another episode of That's a Lot of Science, the podcast for science lovers and everything about science. Today, we will be talking about a very hot topic in medicine and health, which is the microbiome. And to do so, we have our guest here today, Emilie Gladbach, PhD, microbiome pioneer in research and also like microbiome expert, of course. And so first of all, Emilie, thank you very much for accepting to be here with us today. And the first thing I do with our guests is usually leave them a little bit of time to talk about themselves, introduce yourself, your background, so then listener can know you a little bit better. Please.
Sure. So thank you so much for the invitation to join this podcast. I'm very honored to be talking about the microbiome, which I think we will come back to a bit later what it actually is, but it's an area that I'm like really fascinated about, and there's so much progress going on. And I think that like in the future, we would also need, like when we're looking at developing drugs, we cannot just like bypass the microbiome. We really need to look into this. But I think like going a bit back to my background, that is like I think since I was a kid, I was extremely curious. I was like looking under, like I was digging up roots from the trees and wondering like how they got water. I never really, I think at that time, found out why. But all of these different experiences I made while I was a kid kind of led me to study biotechnology at the Danish Technical University, which is quite of like a broad degree. Like you get a lot of different insight into different disciplines. I think for me, the human health and disease was probably always what I really liked. And then I think at some point also working and doing projects with some collaborating partners at, for example, the Danish Serum Institute, which is a reference laboratory for example, different microbial strains, but also they also do vaccine development. I got really like into this area of both like developing, things and in science, and also they were front runners at that point on how to actually do the methodology for like a reference lab. Yeah, and then I think where I really found out that I had this big love for the microbiomes and for the parasites and for everything else that you have in your gut is when I went to this university in Australia for a semester abroad. We had a lot of epidemiology, and as you probably know, in Australia you have a lot of different diseases than you have in Denmark, like parasites, and like we have all the worms, and you have bacteria that can kill you, or actually at that time it was more than here. And they also put a lot of emphasis on it. And then another thing about this was that I really like the human part of it, where we actually in the anatomy class it was applied anatomy, and we got to cut open the brains, like really, and look at all these vessels. And at that time, I was already like imagining like how does everything work. We didn't know anything about the gut-brain axis at that time, but we knew some. And I think if you then go forward in time, I did actually leave academia for a little bit because I wanted to see what it was like, on the other side. So when I was 26 years, I got to become a laboratory team manager, managing a lab and also some people, which was a bit scary in a stem cell company, which is of course far away from microbiomes. But basically, we were harvesting stem cells from the cord blood of mothers who just gave birth. And the idea was that we could give like if these kids or their siblings got sick, you could use these stem cells, for example, for cancer therapy, which I think is an area that has, of course, evolved a lot since then, and is much more, well known. Yeah. But then at that time, that company was to be bought, and at the same time, I put in some applications for a PhD. There was a few people that said you should do a PhD, and I was like, yeah, sure. Yeah. And then I was so lucky that I got into one of the Novo Nordisk Foundation centers. This one was at Copenhagen University in Copenhagen, and focused mostly on metabolic research, but not just on the human genomics, also on the microbiome. And I entered this group, which was almost new. We started up the lab, and our only focus was actually the microbiome. And at that point, it was just the microbiome as in bacteria. But we did make a proposal, and that was to look at more, bacteriophages, which is basically, the viruses that goes into a symbiotic relationship with the bacteria. And it was really, I think, an extremely exciting time there, and where also the whole center that I was in grew from being maybe 40 mostly medical students and medical doctors into maybe 400 or 500, with more transition into like bioinformatics. And the group I was in, and I still connect a lot with, was a systems biology group, and really with a lot of different competencies, and not just people that were doing programming. We also had a lot of people going into the lab, and I think this mix was so exciting. I was like really buzzing, and we always got to go to nice conferences. So
Nice.
and since then, it's not that I didn't like academia, but I want to see again. I'm a bit adventurous to see what it was to be on the other side. So I was lucky to get a position at a startup or established startup in Copenhagen, working with CRISPR. So basically, CRISPR is where you can edit, for example, genes or genomes, and get, for example, bacteria or other organisms to produce other things, or to change like their mechanisms. And there I stayed for a little while before I joined, actually, so this was pharma, but then I actually joined more the nutraceutical side of things with Novozymes, which is now not Novozymes, but now merged between Christian Hansen and Novozymes into Novo Nordisk.
Okay.
And that was really a good time. I feel like I learned a lot about developing like nutraceuticals, probiotics. I think we'll get to that later. And the whole innovation pipeline, really realizing how important it is, like, to check off the different steps before you can actually use the microbiome for anything, like regulatory, and look at the IP and things like this. Yeah.
Amazing.
Yeah. And now?
Now? What are you doing now?
Yes, so now I have exited Novo Nordisk. It took a few months to figure out, or actually to be bold enough to start a company of my own, and actually also starting a company with one of my former colleagues and a few other people. And we are doing consultancy work, mostly within the whole innovation pipeline. So everything from like looking at the scouting and fitting it to the market, and looking at IP and regulatory, and all the way to clinical, and also to launch and commercialization. And I'm very excited about that. And then and then, as I said before, I really like to do a lot of different things.
First of all, congratulations, because your background is super impressive, and you have done so many different things, and you touched upon already a lot of different, I think, crucial part of being a scientist, being curious, being adventurous, moving around the world. You say you were in Australia, and you also were thrown to very responsibility role at a very young age, because 26, I mean, can be scary, as you said. So also, like, the uncertainty. So already, like, I mean, I think I can resonate a lot with that, because we as scientists have been through all these stages, and we have been questioning ourselves. Are we doing the right thing?
But I think, isn't it nice? Like, I mean, isn't it nice to be thrown all these problems or all these things?
Exactly. So
Yeah,
that's one thing that is very important to remember, like to keep
yeah,
always this, challenging mind, and make sure that you're
exactly.
open to challenges and you
Yes,
welcome them, and then you
yes,
can grow from it.
exactly.
As you are a great example of it, and you move from
Thank you.
academia to industry, back to academia, and you now starting your own company, so you are challenging yourself
Yes.
every day, and that is the greatest part of being involved in science in general. Now, I mean,
Yes,
you
yes,
specifically
definitely.
focus on microbiome,
But I
and
think, and I think you also really need to be very agile in the scientific environment, right?
yeah,
It's also very competitive, and it's not necessarily that I like this part of it, but it is competitive,
that's
and so of course you can have a lot of great ideas, but you also like need to figure out how to, how to proceed.
yeah, exactly. I mean, you have to find the right path
Yeah.
for you, but also for what is the needs of the scientific community, of the society, and make sure that what we are doing as a
Yes.
value to the people that is going to use it.
Exactly.
And okay, well, I mean, thank you very much for this introduction. It really gave
Yes.
us a lot of talks point. And okay,
Yes.
let's start with the topic of the episode then. And
Yes.
so, what is the microbiome for those that might not be very familiar with the topic? I know it has become a really hot topic
Mm.
in general in medicine and science. I've been working for a couple of years in a nutraceutical company as well, and I've been working with microbiome as well. But I mean, what exactly we mean with this? Why it is so important as well?
Yeah, I think it's a very good question, and I think the first answer is that we are surrounded by microbes, and it's not just on our body; it's also
Mhm.
like in the whole environment. I would say like our Earth is also a bit founded on all these microbes, right? Starting with viruses and then, yeah, becoming what we are today. Um, and I think the latest numbers for how many microbes that actually or microbial cells is it that cover our body is about 39 trillion microbial cells, which is it's a lot, right? You can't
Yeah,
even think like, how
yeah,
many zeros is there actually on that?
yeah, exactly. And also,
Yeah.
always fascinated me that there are more bacterial cells on our body than human cells.
Yes.
So
Yes,
we are
definitely.
basically more bacterial than
Yes.
human.
Yeah, yes, and I think also when we talk, like I think most people have an idea now what is a microbiome, but when you talk about it in like society or with dinner with people, nobody really thinks about the microbes that are not in your gut, right? It's we are more talking like we know a lot about the gut microbiome because the gut microbiome is diverse and the gut microbiome is shown to have associations to all kinds of different disease and health outcomes. But actually we have a lot of microbes on our skin as well, and we have a lot of microbes in like in our mouth and in the vaginal tract, and like we have everywhere, and they can be so important for example how we feel in our immune system. But I think we're getting back to that bit later. And um, and another thing that's important is that when you think about the microbiome, we think about bacteria, but it's not just bacteria, right? Like we think about bacteria because that's one of the things that's more easy to measure. But we actually have a lot of these bacteriophages that I mentioned a bit earlier. We have a lot of fungi also in our gut. And we have a lot of archaea and other types of microbes. And all of these are like extremely important. And if we go, I think if we go to the very beginning, that's where I always a bit, start when I have to tell people about what is a microbiome, is that where does it originate from? And of course, there's something about when mothers give birth that some microbes, like from the skin and other places, can be a part of their microbiomes. Um, but in the first in the part of the infancy, like just when you're getting out of your mom, you are like getting born either vaginally or you get born with a C-section.
Mhm.
And at that point, it has a lot of impact on which microbiome is actually being developed. Like you will have a lot of bifidos, for example, in the start, and then you'll have some lactobacilli. But all of these events, also whether or not you get breastfed or not, or whether or not you take any antibiotics or other medications, um, can really change your microbiome. And why that is important is that this microbiome development or maturation actually goes hand in hand with immune maturation. So I think if you imagine a bit that if you're born and then you have a lot of these different negative events that impacts the development of your microbiome, you will later in life be more prone to getting disease, and especially like allergies is like something that has really been proven by a lot of different researchers. But also you'll be more prone to getting potentially like also like other like brain developmental issues. It doesn't have to be severe, but it could just be a little bit, and other things like that.
That's one thing. I mean, yeah, you said it rightly. I mean, we always think about the microbiome as only referred to the gut, and also like
Exactly.
the type of products that's been sold until now with like probiotics and stuff. They always claim something related to the gut, but that's
Yes.
not just about it. I mean, you mentioned already the immune system and also the gut-brain axis and how the health of our microbiome, not only the gut, but all the microbiome can impact also the release of neurotransmitters and in turns changing the way our brain function, and we know how this is important. So, I mean, the fact that we are covered in these microbes that fungi and other microorganisms that you mentioned before, but the fact that they are so intertwined with what we are, it's really fascinating. And that's like, why it is important that we take care of it, right?
It's almost like I always think about it as an extra organ, or it's more than an organ, but that's like how I think of it. It's also the fact that the microbiome also have a huge role, for example, in digestion. So it's like the microbiome is like all the bacteria, so the genomes and the functional potential also of the genomes can, for example, produce things like short chain fatty acids that are really good for your like your digestion, and also like propionate and acetate and all these different compounds. And it kind of also reflects like how do you feel, right? Because if you feel good in your stomach, the chance is that you feel good in the rest of the body and in the mind.
Absolutely. So and that's then gives me another question. So how do we differentiate between a healthy microbiome and a healthy microbiome? So I mean, the people might not be aware of the fact that, as you say, there are different species and different types of microorganisms that can impact. So what someone can do to differentiate between the two and make sure that he has a healthy microbiome?
So I think first of all, I will say it's like this differentiation between a healthy and an unhealthy microbiome is not that easy. It's you can definitely say that in microbiome research that it has shown that you have, for example, some microbial signatures in your gut that's related to, for example, liver diseases that I did a lot of my research in when I was doing my PhD. But on the other hand, it's really hard to say then when you have a control group for this other unhealthy group, what is then their healthy microbiome? It's not very easy to define, but you can of course affect your microbiome yourself through diet, for example, and exercise and sleeping well, and potentially also winter bathing, like we do in Denmark. So that is but in principle you want to have you always said for a long time that you want to have more diversity, so more microbes. It's not always what you want, but in theory it's what you want. Because for some diseases it can be different. But then you, for example, want your microbiome to be really good producers of different metabolites that you can use for your body, like we talked about before, like digestion. And it could also be many other areas. So that is, it's hard to define what is healthy, but you can definitely affect your microbiome.
Exactly. So exactly. Now also because, as you said, we are all different, and the diversity microbiome doesn't mean the same thing for every human. So some people might need more of a specific
Yes.
metabolite or a specific, byproduct of microbes
Yeah.
rather than another. And this is important because sometimes we feel like we are not we are considered as a whole, like as a whole species, and the product that you find are designed for everyone. But I mean,
It's not.
I think
Yes.
in the microbiome research, it's crucial, the precision medicine and the targeted medicine in this area.
But I also think like when we talk about like healthy and unhealthy, it's maybe also more thinking about what do the microbes actually do that's in your stomach, right, or in your gut. And that is really like if they can prevent, for example, pathogens to enter into the bloodstream and cause liver damage or other things, then that is also kind of a healthy microbiome, right? And if it can support the immune system, and if it can create like a metabolic balance, for example, where it can help absorb nutrients, that is a healthy microbiome. At least that's how I would be defining it. And then, like as we talked about, the diet would influence the antibiotics, and also generally the lifestyle, right? There's a lot of different things that influences.
Yeah, also because exactly, you can influence it in so many ways. But we sometimes forget because, of course, our sleeping routine can be a little bit crazy sometimes. We are stressed at work. We don't do exercise, or I don't know. We have just been over the Christmas holiday, and we know how these are.
Oh yes. I think we need to eat some more greens in January.
Exactly. Go back to a more balanced diet after this.
Yeah, it's not always just white January. It's like green January.
Exactly.
January.
Colorful January. And moving into then the research in microbiome. So like how you have done this in all your, throughout your career, but how is this conducted? So usually the point is, for what I've saw, is to find always like novel species or novel strain, which is a subspecies of a specific microbes. And how does it work? Where do we start, and how it is conducted?
So I think in order to talk about that, I think we should probably talk a little bit about some of the breakthroughs that actually has happened in the microbiome world. And some of them were quite defining for actually my research career, because like for the past decade or a little bit longer, maybe almost two, there's been a lot of new tools that can help you go through like a whole lab and data pipeline to recover microbiome data. And that is something that Illumina actually more or less not started, but was actually really the front runner of. And that was initially back in 2006, so that's some time ago. And at the same time, they were kind of developing these short read sequences. So basically, you can get all these very short reads of bacteria out, and then you can connect them together. Then there was another company called Oxford Nanopore, and they were doing these long read sequences, and more like, I remember we had these some portable ones that we got for free during my PhD, and they were on our desktop, and we tried. We never really at that time succeeded. We did later when it was more up to date. And then at some point in this like time, like maybe in around 2016 or around that time, maybe 14, we also had much more this change from having only looking at a particular part of the genome in bacteria. So for example, this part is could be 16S rRNA gene, into looking actually at the full genome and being able to compile these different reads into having more depth data. And then I think now we can jump in. Now we can start from where what you were asking originally, which was how do you actually do this? And you, of course, have some kind of sample. It can be, if it's the human sample, it can be like a fecal sample. I put my hands in a lot of fecal samples during my PhD. At that time, I would say it was not so common to talk about feces like over the dinner table, but it became very common in my home. And yeah, and I also think, you know, when we went to conferences, and it was just quite normal to talk about this content. But then it could also, of course, be like skin microbiome or like nose or oral, which doesn't have as many microbes. So you have a sample, and then you need to, of course, figure out which extraction kit you need. And the sample also needs to be quite cold. Should be like either directly coming from the source, which is not often the case. So it should be maybe around in Celsius 80 minus 80 degrees Celsius. And from this point, then you can start to extract. But there's so many differences in protocols. So I think that was also one of the things that I was doing while I was doing my PhD, was to compare some of these protocols for fecal extraction and like for the whole pipeline, which was interesting because some of these protocols, they create a lot of bias, and others of them doesn't create so much, but maybe then they have other things that are maybe not optimal. And then you can, after that, you depending on what you do, whether you do the 16S or you do the shotgun sequencing, then you need to go some different ways and build libraries of your sample. So it's like the metagenomes of like all the bacteria, you want to capture them right as good as you can. And then depending on the sequencing depth, then you can capture either, you know, not so many or the top ones that are abundant, or you can go deeper. So, and that is actually, that's quite interesting.
And that is where you find the very, you know, the new strain, the new, you know, mutation that might have some interesting properties, for example, no, some new features or something that is different.
Yeah, so I would say that you can get an indication from metagenomes, but then you need to, of course, go out and do like the full genome sequencing on a specific microbes, and for that you of course need some qPCR as well, some PCR primers that can amplify what you need. And it can be tricky, especially if it's not very abundant. And actually, many of our keystone species in our gut are not very abundant, but they're very important for the function of our body.
No, even if, exactly, even if they're not abundant, they might have that specific role that no one else covers in your body. And then, I mean, that's maybe one of the biggest challenges, right? I mean, finding the perfect extraction method to get all the population that you need, but also what do you think is going to be? Because of course, like once you identified, for example, a strain that might be potentially useful, I mean, how do you develop that into something interesting, into something useful for medicine, for example?
So, so of course, there's a lot of different steps. I would say, like you can. One thing is that if you have an idea about what is the target, then you can better screen. So if we are talking about now we talked about the gut-brain axis. So if you are talking about the target is, for example, to close, like to keep the barrier in the gut, like to keep the tight junctions tight and not open. Then it could be like that is a target, but then there could be specific targets within that, and then also for the vagus nerve up to the brain. And that is, of course, on the science side. And being now also trained in industry, it's always, as I know from my my previous colleagues in business development and the commercial, like important. What do you need in the market, right? That's the first step. And then you go beyond that, and then you can look at the scouting, and you can look at so like having different types of strains that you want to look into, and you can also look a bit into like this whole IP area. What is a freedom to operate, for example, within the field. is there difference between the US, EU, India, Australia?
Yeah, so usually it's like, yeah, exactly. You have to keep in mind, many things, but in particular, like exactly what can be the application of
Yes.
my, in terms of health of my microbe, and then I look for specific strain that might have the features that I want.
Yes,
And
now it has become more easy to look at genes, for example, and also the function, because we don't, like, I would say it's important, of course, which bacteria are there, but it's more important what they do, and whether, like, sometimes if you design a candidate for could be like a gastrointestinal disorder or whatever, then you have it, but maybe it doesn't reach the gut, or maybe it reaches the gut, but it doesn't have any effect, or maybe it just gets degraded. And these things are also very important. Like, what do you need to code it? Is it more like it could also be? Is it a cream? Is it like, what is it? Yeah. And then of course, always keeping in the back of your mind, what do you want from the clinical protocol? Because I think I've seen, and I also did this a lot when I was doing my PhD, is that you look a lot when you're a scientist, you have a tendency to only look at the preclinical part, so basically where you do the in vivo and in vitro, which is of course great, and in many instances you can use it, but sometimes it's not translatable to a clinical protocol. And that you really have to think a lot about.
I totally agree with you. I mean, I have worked, I mean, this is not a problem only with microbiome. I worked with small molecule drugs, and there is always this problem of translational between the preclinical setting into the clinical, because I was more expert in the preclinical side, and you don't really think about it, but you just make new molecules, you test them, and if they're active, you think you are successful, you're going to be successful, but that's not always the case. And yeah, I mean, it is important. I mean, any time, but yeah, I mean, in microbiome research, but also in small molecule drug research, to always remember that the final aim is to go to humans. So since the very early stage of research, keep that in mind, and design the experiments also for that specific, final target, which is the human health. So that is very, very important.
That is so true.
Yeah. And you mentioned already a lot of very hard technique like genome sequencing, qPCR, but I mean, what's going on there? I mean, do you see anything new coming on? There is any novel technology, or I mean, I'm moving towards the AI science with my company, and there is a lot of data coming from here. I imagine. I mean, how are these technology helping? Are they helping? Do you use them? Is it something that you have seen? I mean, just hype, or there is an actual need for that?
Yeah, I think so. Now we mostly talked about the whole lab pipeline, but there's the whole computational part to it as well. Of course, when you have the data, and it's a lot of data, it's not just a little bit of data, you of course need to sort the reads, and you need to figure out how to sort them. And sometimes it's of course based on your, like, what you have done before and what people do, but there's no really good standardization in this field, right? And then you need to define the taxonomies, and then you go on from that. And I think AI, as you mentioned, will and is definitely a game changer, because these microbial communities that you have in your gut, they're very complex. And that means that you, like, all these trillions of microbes that you will get out as sequences, like a lot of, just get out this black box of sequences, and then you need to look into it. And there, I think that both like ML guided machine learning is is really, really important. And also like just essential in this case. And it's not just about like just cataloging the data, but it's also about like, we could think about like predicting how could these microbial communities or specific microbes actually influence our health. And in this way, we could actually use it for developing a more type of personalized medicine, right? And I think that would be, taking this black box and using it for something that would be.
Yeah, exactly. I mean, that's one of the use application that we can see for it, because of course it will allow you to not only analyze big data, which is definitely something that we can do using this technique, but also like use it to design a new one. And about designing new microbes, you mentioned already CRISP, which is a gene editing technique that it can be used also for microbe design. Do you see that being used? I mean, probably CRISP is a big topic that would need a podcast episode on its own. But what I mean, do you see this also being used in this particular area?
Yes, it is definitely being used by different companies for different targets. It's of course not being used in the more like food side of things, like nutraceuticals, but it is being used in pharma. And it is like, if you think of it, it is kind of ideal if you can engineer either like the box you already have to, for example, produce some substances you could use to maybe increase your production of serotonin or other things that are important for your body. It's not just being used for that, but like could be for the phages as well, the bacteriophages, if you want to target some pathogens better. So yes, I definitely see it as a very, very good tool to use.
It is used also, like, I mean, it seems that it's getting, a lot of attention also, as you said, from in pharma companies, because it's always been referred to as an academic tool, something that you use for basic research, or meaning like academic research. But it has actually application also in an industrial level. And on top of that, like, are there any other technology that you want to talk? I mean, you have something that you come across and it's interesting to talk.
Yes, I think that of course all these different, like now we talked a lot about sequencing, and sequence is basically to get the DNA, right? Here you go. But the DNA is also in principle dead, right? Like you get it out. If it's from a fecal sample, you get it out, it's dead. So it can be a capture of yesterday, or it can be like a capture of, so it's not always what is necessarily most precise. And I think that having all these different omics, for example, you have metabolomics, can tell you more about actually the metabolites you produce. You have proteomics, tell about the proteins that produce. You could also look at more at the sequencing the RNA, which is more of a snapshot. It's not always easy. And integrating some of these layers, and then of course some of them will not be used because they overlap. And then I think we already talked about AI, but I think another thing that is quite interesting is all these organoid models, like these lab on a chip models that can be used, because we are kind of in need of better models than animals. Because if you think about it, a lot of the time when the in vivo part, so that's where we use animals, is being done to translate it into something that could be like close to clinical. Then you have these animals that can be germ-free, for example. You have animals that have maybe a few more germs, and you have some different categories. But the thing is that the immune system is never matured. So all of these alternative techniques, we also had this SHINE model in the group I was, which is basically like a gut simulating model where you can put your samples in that you want to test, and then you can take samples from it from the different parts of the gut. That is very interesting, but of course it should not be used as a direct translation to clinical. It's more of a, this is what is going to happen if we put this microbial community in here and we add these ingredients, but then at least we have some idea.
Exactly. It's I mean, validation of model is always a tricky one to face with, and it's important to face it at the very early stage, but at the same time it's very hard to do it because it costs time and money and you don't know where you're going. You mentioned organoids. Can you explain a little bit what they are?
Sure. Yes. So basically, it's you want to mimic an organ by having an organoid, and the gut is not necessarily the most easy thing to mimic. But then, in the principle, what you do is that you have the like different cell types you need, and then you sort of have a grown organoid. And most of the time, it's not something you do yourself in your lab. You have a service provider to do that. But there, for example, you could test. Is this candidate for this target? Is it safe for human use? You can get an idea, for example, or you can also model what is going to happen if I do this, then you will have this outcome. So it's basically like having another type of model organism, like a mouse or other. Yes. But I think like when we talk about these mice, one thing that is actually quite interesting is that when just before I started my PhD back in 2016, there was a lot of, or there had just been a lot of publication from some very prominent labs in the world, where they transferred feces, so like doing a fecal microtransplant from an obese mice to a lean mice. And what happened was, and that was quite incredible at that time, is that the lean mouse became fat. And they also did the reverse experiment, and the reverse thing happened. And I actually think that this FMT world is quite interesting, and there's a lot of people now, especially in the Clostridium difficile world, trying to or actually now having approved therapies within the C. diff world. So I think that is amazing. And moving a bit beyond the mice.
You can think, you can think then about the impact that the gut microbiome has on what we are, because actually, I mean, it also dictates how we convert food and nutrients in our body, how we absorb and metabolize them. So it is very important. And you already mentioned, exactly, a lot of techniques now that are used to start like translating the research that is conducted, like in identifying strains, identifying novel species, into then therapies. And Shime is one of those like examples. So this equipment that mimic, let's say, the digestive system. So then you can pick sample from any step of the process. You mentioned organoids. And what is then the development moving forward? Like, how are these then translated? Because I mean, we already mentioned the challenges of translate these into a real world therapy, but what is the next step? How it is from other aspects as well?
So you're thinking how it's clinically validated, or are you?
Exactly. How we do then? Because then, of course, with this data in hand. I mean, first of all, talking with microbiome, we usually talk about nutraceutical, we talk about food supplements, so it is different than developing a drug. But still, as you mentioned, like clinical validation is still present there. But also from how you decide what type of formulation is best, what type of product is best for the type of microbe or microorganism that you have in mind, that you have discovered.
So I think it's a very important point, and you of course have you have to come past some ethical approvals and also conquer some regulatory landscape to get into this place. And like when we talk about the nutraceuticals, we still want to do some, especially for a human, we want to do a few clinical trials, testing safety, for example, and also efficacy is really important. And then you can more or less move on with this product, whereas live biotherapeutics are different. And these live biotherapeutics is basically like developing, like you would do in your former career, right? With these small molecules that you need to go through the whole mill of clinical trials, first starting in healthy humans, then you move on maybe to someone like a target population that is not extremely sick but can still be sick, and then you go on, and if it, for example, was cancer, then that is the last group that you would test it in. Yeah.
So in that aspect is very similar from the, at least for the clinical validation, is kind of similar for what happens with drugs, now that you have to go through the different steps. Are they a little bit, I wouldn't say easier, but like shorter in terms of timelines? Are they less stringent than drugs, given the fact that we sometimes talk about clinical validation, but not about claim against, a specific disease? Or, of course, the level of quality needs to be there, but in terms of regulatory as well, it's different.
Yeah, it is. It's of course compared to the rest of the pharmaceutical world, it's the same you need to go through. It's not necessarily easy. And one thing about the microbiome world that we touched a little bit upon a little earlier is that there's not really a good standardization. And that also means that when you talk to authorities, they are of course very interested in the data that you have, but they're also very interested to get more education, for example, on how do we not only look at the sequencing data, but how do we actually also look at the metabolomics data? Like, how is that important? That was a big theme some time ago, and I think now moving into more of the other omics layers. And then, as you said before, looking at the claims. It's of course, if you develop a nutraceutical or a cosmeceutical, there are other ways of having these claims, because you can't really have real claims. It's more like a soft claim about, could be for skin, for example, that the product that you use could enhance the like the skin barrier through, for example, producing more moist in the skin. And then if you had like a pharmaceutical, you could target more the exact mechanism. For example, if it was an inflammatory pathway, you could say that this that we could claim, or if we can prove it through our studies, that we are blocking the toll-like receptors from the pathogen binding. Then there could also be something where you're actually targeting the inflammation pathway, right? So it's
It depends. Yeah, exactly. It depends on the target, on the things that you want. I mean, the microbiome and in general, like microbiome products, can be, as you said, classified into many different things. I mean, food supplement, nutraceutical, cosmeceutical, pharmaceutical products as well. So, I mean, depending on which way you are going with your product, then of course the regulatory and the path that you have to follow changes. And you say that there is an education side to the regulatory as well. Do you have you seen that from the regulatory bodies there is a growing interest in this sort of science, or I mean, what was your view on that?
Yes, so there is definitely a big interest from the regulatory bodies, and it's not like, as you know, that EU, for example, is not as easy to get any drug through that it is in the US or other markets. And I think that it's just really good that they're really listening, and there are also some big fora in this microbiome world that has really taken on the task to push these a bit forward. And there's more and more actually organizations trying to do this, because there are a lot of different like regulatory hurdles within this. It's a bit like there's not like one framework, right? It's a little bit, I wouldn't say case by case, that's too extreme, but it is a bit not always so easy. And there's also something about, and I think that's really important, that you really, like, one thing is that you can prove your primary endpoint, which could be a maybe reduction or in the target that you want, like maybe a reduction in BMI or whatever.
Yeah.
But you also somehow need to show how is it that you don't hurt the rest of the microbiome. Because as we talked about in the start, the microbiome is so important for the rest of our body. So if you have too big an effect on the microbiome, that's not something you can, from a regulatory point of view, kind of say that it's okay if this drug or this nutraceutical is doing what it should, but it's ruining the microbiome, or it's creating maybe a different like cluster of microbes. Like you really need to keep like play the bait safe, and that's where there's no really good rule. But you really need to have these in place in secondary or exploratory outcomes or endpoints.
Yeah, absolutely. Like it is very important how we design the protocol in the very, very early stage of clinical trial, because then exactly that will dictate the success of your product or not.
Yeah, exactly.
Moving into the product development itself. So, I mean, if you go to I don't know, a drugstore or anything, you would see so many products available, so many different classes and categories. And I mean, you could see, of course, probiotics are the main or like the most known. We know prebiotics, symbiotic. Now there is a growing interest in postbiotics.
Yes.
What are they? How do we choose? And what are they? What are the differences?
So the choosing part is not so easy, but we can start with what they are. And I think now you mentioned yourself probiotics, which is also what most patients, most consumers knows most about. And these are basically the live microorganisms that have some kind of benefit that is proven. Most of the time is proven. Sometimes it's not proven, but they should in principle and theory be really important for your gut or optimize it. It could be providing some health benefits like you sleep better or your mood is improved or similar to that. And then we have the prebiotics, and the prebiotics are not microbes themselves, but they're kind of very important dietary fibers, for example, that can enhance the functioning of your gut, or in combination with the probiotics be a symbiotic. And these ones you can also find in like garlic, and you can find onions and like whole grains. So you in principle, many of the nutrients you intake yourself are prebiotics. But it's smart to put it together with a probiotic, because then in principle you can enhance the effect. And that's also usually what you see in both preclinical and clinical packages, that you will enhance the effect. And then you have these postbiotics, and I love postbiotics. I think they're great because they also, like the probiotics, they need to stay, they need to keep alive, right? So the product is not as, it will be stable, but it's not as stable, and it depends also on which type of strains. You have, for example, some of the strains that are being typically or traditionally used as probiotic, they're not as stable, and then you have some probiotics coming in later, for example bacillus, they are spores, so they are much more stable, but you kind of avoid this stability issue, or you don't avoid it, but you have it not as much when you look at postbiotics, because postbiotics are basically still a fermented microbial strain that has produced, for example, short chain fatty acids that are really important for like your whole, like your gut and your gut health, and enzymes, and could also be antimicrobial products that can, for example, prevent infections. And when they're not alive, they're just it's just much easier in some aspects to develop it. Yes.
Yeah, exactly. I mean, that's one of the things I remember. I mean, touching during my career on the postbiotics word, and I mean, one of the claim, let's say, that they made was about being stable, or like, I mean, not being a live microorganism anymore. So it's much easier to handle, much easier to produce, and but they can have or they usually have similar properties, because they are sort of metabolite or the substances that
Yes.
the microbiome produce, or like sometimes they are fragments of
Yes.
the microbes
Exactly.
itself. And
Yes.
this is kind of again it's a more recent approach, maybe also due to the difficulties, as you mentioned, of keeping the stability of probiotics, and also like makes them easy from a formulation point of view, maybe. And
Yes.
it's easy. How do you select usually a formulation when you want to develop a new product like this? I mean, what are the features and the type of things that you want to look at?
So for the formulation, I'm thinking about whether it's going to be oral or which type
Yeah,
of yes, yeah. So of course it's a little bit well, it's always about the mode of actions, right? So if is it something that you want to target that is in your gut, then I think the oral way is probably a good solution. And then based on if it's a postbiotic, then you of course you can fill it into a capsule. You need some high quality and stability measures, and also figuring out like, can you translate the way you did efficacy studies in the early preclinical to the formulation? That's very important. And then of course you could also have it in a have a biotic in a lotion. It's not as easy to do that with the probiotics, and generally it's not a very easy field. There's also now more and more products within like fertility, so that would mostly be for vaginal health.
Okay.
There's mostly also capsules. For if we talk more about live biotherapeutics, these are also often being used for, this is pharma, of course, for maybe patients that cannot intake a drug. So in that case, you would need to do that subcutaneously. Yeah. You could also imagine that you would have like a solution for if you had an eye, like an eye infection. In that case, it would be like directly in your eyes. It would not necessarily replace antibiotics, because that's not what we want to do, but we want to create other solutions that you can use maybe in conjunction to antibiotics.
Yeah, absolutely. I mean, no, that's one thing. I mean, of course, then I mean, for certain thing, you still need to go to drug, but it can help you. I mean, also like if you go under an antibiotic therapy, you know that this is going to affect and damage your microbiome throughout the body, because at the end of the day, they are microbes as well, so they are affected by antibiotics. So it is important that you use in conjunction also a live biotic, whatever you choose, therapy to restore the normal functioning of your body. That's important.
Yeah. So and I really like the antibiotics part. Is also something that I'm really really involved in, and I think it's so interesting, because I think for a long time there was, of course, like we used more and more antibiotics, and it was great, and it was prescribed here and there and everywhere. And at some point we found out, as I said, it also ruined the rest of the microbes, or we found out that were microbes that could be ruined. And I think there was also some early indications of this back in the end of the 1950s, where this was actually also for the C. diff, Clostridium difficile infection, where some scientists found out that when you had diarrhea and you gave them antibiotics, they got more diarrhea. So maybe it was not so good. So then they actually tried with a fecal microbiome transplant, or microbial transplant at that time. It was not called that, but
exactly. That's exactly because they didn't realize that it was actually the microbiome doing most of the job there, and they were just killing it. And about this transplant, is that a technique that, I mean, it is like a technique that is used in the clinic, but for, I mean, when it is used, I mean, it is something that we use normally, or it's something for very specific condition or very specific target?
Exactly. So I think FMTs were like really originally used in China. It was actually in the 4th century, and they used it for people who had food poisoning and diarrhea, and I think probably there could be other records of it somewhere else in the world, but basically they said it was a yellow slurry that they gave to people. And now, much later, then we of course had the 1950s events where you did this, where you tried some FMT where people had some kind of stomach infection. They maybe somehow knew it was C. diff, but they couldn't really prove everything at that point, right? And now today it is being used, FMTs, and for a long time it was mostly used in hospitals, so for like non-commercial products where you could basically harvest the microbiome or the feces of a healthy donor, and so defined by not having any diseases or anything else like you would do for a blood donor, and then given it to recipients that needed it mostly for infectious disease, so like C. diff could also be E. coli. But the problem with that used to be that bacteria are not always so good if you give them to immunocompromised people. So there were a few instances where people died. But since that, it has become more safe, and you can filter sterilize it much more. So you basically select which part of the population you want to give. And on top of that, there's one commercially available kit that's been approved by FDA, or FMT that's been approved by FDA, and that is the one that is now a Ferring company. It used to be, it's also still called Rebiotics, and they made this product called Rebiota, which is a fecal transplant, and it was approved in, I think it was the end of 2022 for use. I'm not aware how much it's being used, but the problem with this is, of course, that it's quite expensive. And if you have things like a bacterial infection, it's quite often reoccurring, and then you need to use it a lot. And there are also other companies like Ceres that are really fighting hard to get their candidate into the next trial. They're doing really well in the clinical trials. They have more made, it's not an FMT, but it's like also within this area of Clostridia, where they have prepared a mixture of Firmicutes bacteria, which are like beneficial for the gut. And then there's a few other companies like Vedanta, and they're also looking at all kinds of other disease. Yes.
So there is still an interest in that, no? Because I mean, you say the approval was only like 2 years, 2 years and a half ago. I mean, it's almost. I mean, it's not something that is considered obsolete. I mean, it's still
no, it's not. But at the same time, there was this like when these scientists tried to take feces from an obese to a lean mouse and vice versa, people thought, and that was like in 2012-13, that now we can change obesity, right? And that never happened. And it's like part of it is that the mice and it's not like really translational to the humans, and it's also just that it's so hard, for example, to have a trial like that, because you would really rely on the human being completely honest of what they ate, and human are not, and we've seen that in many trials. So it's very debated still, actually, whether like such a transplant would be beneficial for obesity.
Yeah, exactly. I mean, sometimes, I mean, the hype is always, a problem in this case, because you think about what, and we are back to the problem of translating these from animals to humans, because then you see something in animal studies and you think, okay, that's done, and we can do it in humans, but it's not always the case.
No,
it's rarely the case.
No.
Okay, well, I mean, very, very, very interesting. And okay, I think we already covered a lot of things, and many different, angle to these topics. And sometimes we think about this as a single thing, as that little probiotic that you buy in the drugstore when you don't feel well. but you explain it
Yes.
so well. How many aspects are there? How many different technique? How many different expertise is behind this? And where do you see this going? I mean, where is the field of microbiome going in the next 10 years or 20 years or whatever?
But it's really going into these, I think, personalized microbiome therapies, not just therapies, also nutraceuticals, right? It's also, I think, in general where the market is moving. And here, I think the whole AI also will be instrumental to be able to also unlock some of the complex interactions, because it's clear that it's not just one microbe that is going to act one way. It's like a cluster of, or the combination of. There's also all these host factors that you need to include, and to have all that data and to get something reasonable out of it, you really need to rely on AI. And it's of course the easy answer, but I think it is also the answer. And in doing this, I think we will be able to foster many more microbiome companies that have a focus on like both prevention and treatment, for personalized issues, or yeah.
I mean, in general, we have this, of course, growing interest in, we overcome the paradigm of one medicine fit for all, and now we are just, and that applies to everything that can impact on human health.
And I think one last thing, and we also touched a bit upon it before, but that is really that on the regulatory landscape, it is like we need much clearer guidelines here. And it's not just like I am commanding that we need it, but there needs to be a good conversation on how to develop it, right? For not just for nutraceuticals, but also for like all these microbiome-based therapies. And when we get that, I think the whole like venture cap side of things would also have much easier time investing and getting a return, which would like facilitate even better therapies and prevention methods.
Makes sense. Yeah, absolutely. I mean, the end, exactly. I mean, whether you are doing it for exactly, a pharma products or a nutra products or a food supplement, you still have to deal with regulatory. So it's important that clear. So then we know already how challenging it is. We cover some of the challenges today. And we need definitely help also from that side in order to not waste time and waste chance of successes in that. And to conclude, I always make this question at the very last. What advice would you give to people that are now starting their career? Maybe they are students in microbiology, or I don't know, young scientists that want to get involved.
So I think there's of course I could sit here and talk about that forever, but I think one thing that I think it's really important is to really stay curious and challenge a bit the normal. Like of course, obey your professors and group leaders, but I think you get the best out of always like trying to be critical about what other people are saying in science, and also thinking some more steps ahead. I think like these days where we have tablets everywhere and it's always some lights from a screen in your eyes, then be a little bit better just putting your phone on silent and just dive into an area that you like, because I think you spend a lot of time when you're young kind of searching, right? It's not just searching for your own personality, it's also searching for what is it that you want to do with the rest of your life. And that's not a thing that is only happening when you're younger. It can happen multiple times, and if you don't dive into the things that you like at that time, I think it will be harder to follow what you want. And I think one last thing is that I have always really benefited from talking to people that are not necessarily in the same area as I am, or doesn't have the same background. It can be both. When we are talking innovation now, also with other companies, I sit on the more scientific side of things, but there will also be someone doing more commercial development, and I think it's important, and also important in the groups at academia. I think my group leader was extremely good at doing this. I think really learned a lot from it. Yeah.
Amazing suggestion. Yeah, these are always good. I mean, we start talking about being curious and adventurous, and we conclude with almost the
Yes.
same take-home message, because it is what it is. At the end of the day, scientists are curious people that want to know how the world how the world goes, how how everything works. So from the smallest microbes to galaxies, that's what we do.
You're absolutely right, and like I also think people really should join this microbiome science movement, because it is like it's been for a long time a little bit on the verge of doing something great, and now we're really soon there, and I think we'll see maybe in 5-10 years a lot of really successful companies.
Amazing. Yeah, I agree with you. It is a topic that is definitely on the rise.
Yes.
Definitely a hot topic. It's good. It's good.
You should go in it too.
I am. I might go. I might go back.
Yes.
I might go back. Okay, then, Emilie. Thank you very much for being with us today. It's been an amazing conversation, and I hope you enjoyed it. And for everyone listening, thank you, and speak soon. Bye.
Bye.
Okay. I'm stopped.
That's a load of science.